Dear students, this is Dr. Anjani, Assistant Professor in Biotechnology. Welcome to e-learning sessions in Immunology. Today we learn about blood group antigens. In normal human erythrocyte cells, on their surface we will find the genetically distinct antigens that form the basis for different blood groups. Karl Landsteiner discovered the ABO system of blood in 1900s. He found two types of antigens on the surface of the RBC or erythrocytes. They are antigen A and antigen B. Similarly, there are two types of antibodies in the plasma called antibody A and antibody B. Based on the presence or absence of the antigens A and B antigens and antibodies anti-sera A and anti-sera B, human blood is classified into four groups, namely A blood group B, AB blood group and O blood group. A blood group person on his erythrocytes, the RBC will have the antigen A, and in the blood serum will find antibody B. Anti-sera B will be identified or will be present. But in case of the B blood group person contain antigen B and antibody A, whereas AB blood group containing person will be having erythrocytes. We will find both the antigen A and antigen B, but in his blood sera we won't find the antibodies, that is antibody A and antibody B here. And one more the blood group, that is O blood group containing individual, they will be having the anti-sera A and anti-sera B, but on the surface of the erythrocyte we won't find the antigen A and antigen B. So individual with A blood group in the on the surface of the erythrocyte we have the antigen A, and in the blood serum we'll find the anti-B anti-sera. Whereas in case of the group B individual, on the surface of the erythrocyte we have the B antigen, but the anti-A anti-sera will be there in the plasma or in the blood serum. But in case of the AB blood group containing individual, we have both antigen A and antigen B on the surface of the erythrocyte, but in the plasma we don't find any antibodies. Whereas in case of the blood group O containing individual, they'll not have the antigens, but they carry both anti-A anti-sera and anti-B anti-sera. Till now, the International Society of the Blood Transfusion recognizes more than 270 blood group determinants, and they led to the around 26 blood group system. Most erythrocyte surface antigens are located on the integral membrane polypeptides or the glycoproteins, or in some cases membrane glycolipids. Most blood group antigens are proteinic in nature, that is proteinaceous in nature, with group specificity determined primarily by the amino acid sequences. And some blood group antigens are membrane transporters. Blood group antigens are membrane transporters involved in the transport of molecules across the erythrocytic membrane. So these blood group antigens are also referred as isoantigens. They are also referred as isoantigens. So these isoantigens are antigens of an individual which are capable of eliciting an immune response in an individuals of the same species. The two individuals of the same species, when you inject this antigen, they raise the immune reaction, who are genetically different and who do not possess the antigen is called isoantigen. Antigen A and antigen B are considered as isoantigen. When the two individuals of the same species are injected with these antigen, it will raise the immune reaction, and hence they are called as isoantigens. So these antigen A and antigen B, or the in general blood group antigens, are membrane transporters involved in the transfer of molecules across the erythrocyte membrane. Now we'll see the composition of antigen A or antigen B, or the surface molecules, how they differentiate the antigen A and antigen B. Usually ABH or the antigens are present on the surface of the erythrocytes. The ABO blood group antigens are carbohydrate antigens, that is, epitopes are the result of the oligosaccharides attached to the glycoproteins or the lipoproteins. So the single change in the oligosaccharide will differentiate either A antigen or the B antigen. So the blood group O was originally thought to lack any antigen, but is now known to possess the H antigen. The Bombay group lacked this H antigen. How this H antigen will be formed is, it has the basic structure, that is the N-acetyl galactose, N-acetyl glucose, N-acetyl glucose galactose attached to the proteins or the lipids. So that is the basic structure. If this fucose moiety is attached, by the fucosyl transferase enzyme, that results in the H antigen. So this basic structure will be there in the blood group antigens. But in case of A antigen, we will find N-acetyl galactosamine. In case of the A antigen, we have N-acetyl galactosamine along with the basic H antigen. Whereas in case of the B antigen, you have the basic core structure, but instead of the N-acetyl galactosamine in B antigen, or the galactose will be there. So that is the difference. So A will be having the N-acetyl galactosamine, whereas the B antigen will be having the galactose. But the basic structure with the fucose moiety is called as the H antigen. So this is the difference between the A antigen and the B antigen, which both are acting as the isoantigens within the same species, which raise the immune reaction within the individuals of the same species. Next is Rh antigens. Landsteiner and Wiener in 1940 discovered the existence of a special type of antigen in the RBC of a rhesus monkey. So this antigen, which is present in the rhesus monkey, was showing the structural similarity with the antigens or the factors present on the surface of the human RBCs. Since this factor was discovered in the rhesus monkey, it was named as rhesus factor or Rh factor or Rh antigen. So this Rh factor was named because This is the human erythrocyte antigen which is present on the human erythrocyte cells, and it shows the similarity with the antigen that is present in the rhesus monkey. That's why it is called as Rh antigen or the Rh factor. Based on this, again individuals will be classified, each blood group individual will be classified as Rh positive or Rh negative. There are several varieties of Rh antigens of the antibody. Usually the common Rh antigen will be designated as antigen D, and its antibody as anti-D antisera. It is commonly we regularly use it in the human blood typing, that is anti-D antisera. It is specifically raised against the antigen D. The production of the antigen is controlled by multiple alleles. So more than one allele is involved in the production of the antigen. Rh antigen has no natural antibody. However, our Rh antibody can be produced artificially when he receives the blood from the Rh positive person. When he receives the Rh positive person. Even a small amount of Rh positive blood can evoke the production of Rh antibody in the Rh negative person. So that is the basis for erythroblastosis fetalis. When a woman, when she is Rh negative, when she conceives the Rh positive baby, in her first pregnancy, during the delivery time, there will be mixing of the maternal RBC and the fetal RBC. When this Rh positive fetal RBC mixes with the Rh negative women or the mother, when she conceives the second time, when she is conceived with the Rh positive baby, that is the fetus, the mother body itself will be killing the Rh positive fetus in the womb of the mother. So that is the best example for the interaction between the Rh negative person immune system with the Rh positive RBCs. So even the very small amount of the Rh positive RBCs or the blood can evoke the production of Rh antibody in the human being. Only few blood cells or the erythrocytes, when they are exchanged between the fetal Rh positive RBC with the immune system of the Rh negative mother, there will be the production of lot of antibodies against the Rh antigen. So this is called erythroblastosis fetalis, where the mother, the immune system will kill the fetus in the womb itself. That is due to the Rh antibodies which are raised in the first pregnancy or during the first delivery will be killing the fetus in the womb of the mother. And this antibody, once it is formed, it remains in the human body throughout their life, Rh negative person. So that is regarding the Rh antigens. Next, coming to the blood transfusion. Now we know we have the O blood group, we have A blood group, and then we have B blood group, and then we have AB blood group. So whenever we undergo certain surgical procedures, it is very important to know the blood group of the individual. If the person is O, if the person is having the O type of the blood group, he can donate the blood to O blood group containing individual, A blood group containing individual, and B blood group containing individual, and also AB blood group containing individual. So that's why the individual who are having the O blood group are called as universal donors. They are called universal donors. Whereas the individuals who are having the blood group A can donate their blood only to the person who are having blood group A, or they can donate the blood to the AB blood group containing individual. whereas B blood group individuals can donate the blood to B blood group containing individual, and similarly like our A blood group, here also the B blood group individual can donate the blood to the AB blood group containing individual, whereas the AB blood group containing individual can donate the blood only to AB blood group containing individual. But when you check the chances for the AB blood group containing recipient individual, they can receive the blood from O, they can receive the blood from A, they can receive the blood from B group containing individual, they can receive the blood from AB blood group containing individual. So that's why AB blood group containing individuals are called as universal acceptors, or there where you can donate the blood to AB, all the blood group individual can give the blood to AB blood group containing individual, but AB blood group containing individual can donate only to AB. They can receive it, but they cannot donate to other groups except AB. So this is very, very important. At the same time, when we consider the blood group, each time you should check for the RH factor. So with RH factor, that is RH positive individual should donate the blood only to RH positive individual, and whereas the RH negative individual should donate the blood only to RH negative blood group containing individual. So in each blood group we'll have the RH positive O blood group, RH negative O blood group, and similarly in case of A also you have RH positive A and then RH negative A, and also in case of B you have RH positive B and RH negative B. It is usually simply designated as B positive and B negative. Similarly in case of AB, AB positive will be having the RH antigen, AB negative will not have the RH antigen. So each and everything has to be taken care before we go for the blood transfusion, either donating or in receiving the blood. So this is regarding the blood transfusion, and by learning this, learning the structure or the chemical composition of the blood group antigen, it has very good applications. So like you should know the blood group antigen during blood transfusion, hemolytic diseases, and during organ transplantation. It is very important to know the blood group antigens, and also in solving the disputed parentage, the blood group play an important role, and in the detection of the culprits in the criminology or in the forensic science, it play an important role. That is, blood group antigens or the blood group, the blood cells play an important role in identifying the culprits. So these alloantigens or the isoantigens on the surface of the erythrocytes, they are present in the particular human being or the individual. So this identification of these antigens, isoantigens or antigen A and antigen B on the human erythrocyte, is called as blood typing or blood grouping. The matching of the donor and the recipient blood groups for the ABH, that is A, B, O and AB blood group and RH antigen is essential for the success of organ transplantation and also during the blood transfusion. The transfusion of improperly matched, the blood groups, when they are not matched, they immediately raise the severe or the acute immunological reaction that will be very fatal for the recipient individual. So it is very fatal. Immediately the blood clotting takes place and the person will lose their life. So that's why we should know the blood group antigens and the blood typing. The blood typing should be done very neatly in order to avoid these the accidents, or to avoid these wrong identification may be fatal for the one's life. So in this session we learnt about the blood group antigens and then blood transfusions, and how the blood groups will be classified based on the A antigen, B antigen, and the H antigen which are there on the surface of the erythrocytes. I hope you understood the topic. Thank you.